And the defect I'm studying has naturally very high polarization. Usually in room temperature you have equal probability of being spin up or spin down. The defect I'm studying, if we address it with light, it's always spin up. And you can transfer this, the fact that the defect is always spin up, to the analyte, and you have a bigger magnetic signal from the precessing spins. So that's essentially what I'm doing now in my experiment. I have a big magnet, the diamond is somewhere in there being addressed by green light, and I'm trying, yeah, you can kind of see the chip is kind of in a small angle based on the geometry of the defect in diamond. And I am trying to figure out the best way to transfer the polarization from the defect to whatever I'm studying. And that's the gist of my experiment.
Hi, my name is Nate Rissoff, and I work on this project here in Victor Acosta's lab. And so the experiment that we're running is we're trying to measure the new relaxation of magnetic nanoparticles. And so the basic idea is this, that we have a little nanoparticle, and inside of it there's this tiny magnetic moment. And after some characteristic time called the new relaxation time, the magnetic moment will flip.
So hi, I'm Brian, and this is the experiment that Nate and I work on. This is one of our lab's magnetic microscopes. And so many parts of this are similar to many fluorescent microscope setups. We have an excitation laser that comes in here through the objective and illuminates our sample. And that same objective collects the fluorescence from our sample and is imaged by a camera over on this side. But the difference in our microscope is that this is used to image magnetic fields. And the way we do that is we mount our samples on top of a small diamond chip that's doped with some defects called nitrogen vacancy centers. And by driving those NV centers with microwaves, we are able to look at the changes in fluorescence produced by the NV centers and relate that to the local magnetic fields. So that's how we image magnetic fields.
Hello, my name is Forrest Hubert. I'm in Victor Acosta's lab, and in this experiment our goal is to use atomic defects in diamond, the nitrogen vacancy center, to image magnetic fields of small particles. To do this, what we do is, our goal is to take super resolution images. By super resolution, what we mean is we're going to use some characteristics of the NV center to bypass the diffraction limit of light.
Hi, this is Nazanin, and I'm working in Victor Acosta's lab. So Forrest and I are building this setup. He actually explained what generally we're doing, and what we build is a modified confocal microscope. And you can see it's a multicolor confocal microscope. So we have green, we have two red, this is fiber coupled, and we get red out here, we get another red here. And so let's just go over the setup. So we're using a VPP. This is the VPP vortex faceplate. And what it does is that it's a different, so the beam goes through different thicknesses of glass. And then eventually we're gonna actually generate a donut beam through that like interference pattern that they actually destructive interference. So that makes a donut. And then we have cool features. So the reason why I call it a confocal is because of this pinhole in the emission path, that we get rid of all the light from other different depths, and we only collect light from a single layer depth of the sample. So we're here mounting our diamond. You see the objective and a tiny diamond on top of it. We need the piezo stage to move around tiny amounts on the diamond. And what else I can tell you guys? There are so many things, but I think that's enough for now.
Hi, I'm Max Aiello. I work on the Metasurface project. These are optics that have nanostructures imprinted on them. In the past, we've made structures in order to make a varifocal lens using this process. And now, what we aim to do is to make a multi-layer achromatic lens. This is done by, as you can see here, we run an FTTD simulation in order to characterize our nanostructures, and with this information we're able to translate that into a design for our metasurfaces, and then in the clean room those metasurfaces are fabricated and tested on our optical table.
Hello everyone, I'm Yasser Solani, a 6-year PhD student in Victor Acosta's lab. Here I'm doing ACDC magnetometry using NV colloxamazine diamond. In order to push forward the sensitivity of the diamond sensors, I am adding fried cones in a bolty shaped manner. These guys. What they are essentially doing, they are collecting the flux, magnetic flux from large volumes, let's say centimeter cube, to a small volume, millimeter cube, and where our diamond displays it. In other words, they are enhancing the magnetic fields around the sensors by a factor of a few hundreds.